Good afternoon, dear students. Lecture number 8. The topic of the lecture: features of human population genetics. Plan of a lecture. Evolution is a population unit. Characteristics of the human population. The next one: features of the gene pool of the person. Hardy-Weinberg's law. Conditions for the manifestation of the Hardy-Weinberg Act. The supporting genetic equilibrium and causing changes factors in genetic structure of population. And the next one: mechanisms that disrupt the equilibrium of genes in the human population. Population and the statistical method and its value for study genetic structure of populations. In 1903, the Danish geneticist William Ludwig Jorgensen first used the term population to refer to a group of genetically heterogeneous individuals. A population is a collection of individuals of the same species living in the same territory for a long time, freely crossing each other and giving fertile offspring. Population genetics is a section of genetics that studies the frequency distribution of alleles and their change under the influence of the driving forces of evolution. Mutagenesis, natural selection, gene drift, and etc. The formation of population genetics was most influenced by scientists Timofey Frissovsky, Четвериков, Kaczynski, Wright, John Holding, and Donald Fisher. The genetic characteristics of the population is its gene pool. The Russian scientist Serebrovsky has formulated this term. A gene pool is a collection of alleles that form the genotypes of organisms of a given population. Gene pools of natural populations differ. Accountable diversity, genetic heterogeneity, polymorphism, and genetic unity. Human populations are characterized by the following characteristics. At first, sizes. Populations are considered large if they have a population of more than 4,000 persons. Populations whose numbers range from 1.5 to 4,000 people are called dams. If 1.5 thousand are isolates. Secondly, fertility and mortality, quantity of newborns and deaths. And the next one, age structure, quantity and ratio of newborns, children, adults, old people. Sexual structure, quantity and ratio of men and women. Spatial structure, distribution people on the different territories. And the next one, environmental state of the environment, nature and anthropogenetic influence. Economic situation of society. And the next one, climatic conditions, nature environment during certain period of population being. Genetic structure of population is determined by variability of genotypes, frequency of variation of individual genes. The genetic structure of population is determined by the marriage system and factors that change the frequencies of genes. The mechanisms that determine the genetic structure of population are divided into two groups: balancing genes, unbalanced genes. Genetic structure of population can be studied with the help of Hardy-Weinberg law. The method is based on a pattern set in 1908 by English mathematician Gottfried Hardy and German physician Wilhelm Weinberg for an ideal population. The pattern they discovered was called the Hardy-Weinberg law. Wording of the law of Hardy-Weinberg. In an ideal population, the frequencies of genes and genotypes are in equilibrium and do not change in a number of generations. The formula of Hardy-Weinberg law is P plus Q equal 1, or sum of P and Q to the second power equal 1, or P to the second power plus 2pq plus Q to the second power equal 1, where P to the second power is frequency of homozygous genotype occurrence by dominant allele. 2pq is the occurrence frequency of heterozygous genotypes. Q to the second power is the frequency of homozygous genotype occurrence by recessive allele. The Hardy-Weinberg law is used to predicated genotype frequencies in population. This law based on Mendelian laws. It is derived from a simple Punnett square in which P is the frequency of dominant allele and Q is the frequency of recessive allele. If we know the frequency of homozygous genotype occurrence by dominant allele in population, we can find the frequency of the homozygous genotype occurrence by recessive allele. There are the factors supporting genetic equilibrium in populations. 1. Population must be limitlessly large. 2. Availability of panmixia-free marriage. 3. It should not be influenced by evolution factors. And 4. Population should not be interrupted in time. The factors causing change in genetic structure of population are mutation process, isolation, gene drift, population waves, natural selection. So, the Hardy-Weinberg law for human population is amended. Wording of the law of Hardy-Weinberg for human population is: in an unlimited large population, in the absence of mutation, natural selection, pressure migrations, the presence of panmixia, there is an equilibrium of the number of genotypes. The elementary evolutionary factors affecting the population gene pool include the mutation process, isolation, gene drift, population waves, natural selection. Well, let's speak about all these factors. Mutational process. Change of inherited material occurs as a result of gene, chromosomal, and genomic mutations. In some populations, there are many people with achondroplasia, polydactyly, for example, but in other ones there are not people with these mutations, or there are isolated cases these disorders. It depends on mutation distribution in population. Mutational process changes the ratio of one allele to another, and the result of hereditary variability forms. The frequency of mutation gene is 10 to the 5th power to 10 to the 7th power in one generation. So 6% gametes carry mutating genes. The dominant mutations appear in the first generation already. They are influenced of natural selection. Recessive mutations more often occur. At first, they accumulate in population. These mutations start phenotypic appear in population only after recessive homozygotes form, and then influenced of natural selection. The saturation of the human population with recessive mutations is called genetic cargo. The genetic cargo explains the appearance of 6% of offspring with genetic defects. For example, albinism. It is an autosomal recessive disease. In most European populations, one person out of 10,000 is an albino. So the frequency of recessive homozygotes is 0.0001. If we use the Hardy-Weinberg's law, we have q to the second power equal 0.0001, so q equal 0.01 or 1%. P equal 1 minus q or 1 minus 0.01 equal 0.99 or 99%. But we know there is 2pq. So 2pq equal 2 times 0.99 times 0.01 equal 0.018198, so about 0.02 or 2%. People with genotype 2pq are secretive carriers of albinism. They contribute to the gene pool, and their genotypes increase the saturation of population with these recessive mutation and distribution disease. This slide is demonstrated how the Hardy-Weinberg's law can be used. The migration and marriage system are also of great importance for supporting heterozygotes quantity in population. In small human populations, population migration is insignificant. Influx of gene from other group was less than 1%, and 90% of marriages are intragroup and relative marriages. As you know, there are marriages of non-related people and closely related marriages. The marriages of non-related people are genetically prevalent in modern human populations, but sometimes in some populations closely related marriages still occur. The consequences of closely related marriages are the appearance of congenital malformations in children, early mortality, and increase in the frequency of stillbirth. The incidence of rare autosomal recessive diseases in related and non-related marriages is on the screen. We see that the incidence of closely related marriages is much higher than in non-related marriages. The next factor which changes the population genetic pool is isolation. Isolation is exclusion or difficulty of free crossing in population. Types of isolation are: geographical isolation is separate of groups of organisms by some obstacle, sea, river, mountain, and etc. As for human, there are the African tribes living in isolated territories. The next one, ecological isolation. The different groups of organisms are in different part of environment. For example, top of the mountain and its food, river bottom and water surface. For human population, different continents or territories with different climate conditions, highland, desert, Arctic, for example. And the third type of isolation is biological isolation. Mutations which prevent the reproduction of fertile offspring. The next factor leading to change in population genetic structure is gene drift. This process was first studied by geneticists Dubinin and Romashov, as well as American scientists Wright and Fisher. Gene drift is random variation in allele frequencies in populations of limited size. It is a mechanism of genetic automatic processes. In certain conditions, climatic, biological, ecological situations occurs. Reduction in population size, reduction in genetic diversity, restoration of population size, but lack of restoration of genetic diversity. It called the bottleneck effect. The number of individuals of populations drastically reduced under the action of some factors. So the number and diversity of genotypes reduced. Thus, there are certain genotypes of surviving individuals of the next generation only. The next factor changing the population genetic pool is population waves. The population waves are fluctuations in the number of organisms in natural populations. For example, when the number of predators falls, the number of its victims, certain animal species, is increased. The one of important factor which change genetic structure of population is natural selection. Natural selection is a process that survives and leaves behind the offspring of predominantly individuals with hereditary change useful under these conditions. There are several forms of natural selection. At first, driving acts with a directed change in environmental conditions. Individuals with these traits that match the changing environment gain benefits. The next one, stabilizing natural selection, directed against individuals with extreme deviations from the average norm, in favor of individuals with an average severity in the trait, aimed at maintaining a previously established average trait to property. And the next form of natural selection is disruptive or breaking off, in which the extreme variants of the feature are preserved and its average values are removed, contributes to the emergence and maintenance of population diversity. Natural selection occurs at all stages of the ontogenesis of organisms. In modern human populations prevails a driving natural selection. About 10% of marriages are infertile. Spontaneous abortions account for up to 50% of conception. Stillbirth to 3%. Early child mortality to 2%. Do not marry 20% of people. Thus, approximately 75% do not contribute to the gene pool of future generations. Different forms of natural selection and isolation contributed to appearance of adaptive types of people. I think you know about Arctic tropical types and highland or desert human type of temperate climate. People with these types have specific biochemical and morphophysiological features which help to adapt to environment. Nowadays, we have a new type of human, a person of the urban environment. There are many people who live in modern conditions of constant stress, high pace of life, and they need and try to adapt to it. In conclusion of the lecture, I'd like to tell about value of population and statistical method. So, population and statistical approach is used to determine or confirm the type of disease inheritance. It provides valuable insights into the identification of factors, pathways, and genetic mechanisms of anthropogenesis. Broad is used to determine the degree of interpopulatory genetic diversity. It is used in calculation of genetic distances between compared populations, which give an opportunity to judge the level of their genetic relationship. Thanks for your attention.